Next question is the why does the insecticidal proteins are not active the bacterium from which it is extracted? How does this protein become active in the larvae of volvorm insects who feed on bt cotton? That your answer is the insecticidal proteins are not act on the bacterium Bacillus thuringiensis because the Bt toxins proteins exist in inactive protoxins. When the inactive protoxin ingested an insect, it is converted into an active forms of toxin due to the alkaline pH of the gut within the solubilize the crystals. The activated toxin bind to the surface of the midguts and cells, create spores, and that cause the swellings and lysing uh, and eventually cause the death of the volvorm insect. Next question: why is the agrobacterium typificans called the natural genetic engine? That is soil inhibiting of plant pathogenic bacteriums. Agrobacterium typificans infect the broadleaf crops including the tomatoes, soybeans, flowers, and cottons, etc. And that's that are utilized for the changing of the genes, so that's are known as the natural genetic engineers. How does enzymes become activated in the larvae of the volvorms? That the enzymes are activated in the alkalines medium of the uh, insects. That in first transgenic cow, the first transgenic cow is belonging to the rosy produce the human proteins and raise milk 25 grams per liters. That milks contain the human alpha lactalbumins and was nutritionally more balanced product for human babies. That's the cows. And the next questions are the why. What do you know about the recognition sequence? That your answer is the showing on your display. That's restriction enzymes in first always cut DNA molecules at a particular points by the recognizing a specific sequence of 6 base pairs. That is specific sequence is known as the recognition sequence for the hen first. That next one is the name restriction and endonucleases with who discovered firstly the restriction endonucleases in the second restriction endonucleases enzyme EcoR1. What is the meant of each letter of enzyme? That answer is the E belonging to the genus, CO belonging to the species, R belonging to the name of its stems, and I as order which enzymes were isolated from that strands of the bacteria. That that answer is that here that strands that are that strand of bacteria. What happened when we ligate a virgin DNA at BAM first the tetracyclines resistance genes of vector PBR 320? That your answer is that here the when we ligate the virgin DNA at the BAMS first, the tetracycline resistance gene of the vector will be lost.